Hello, it's Mr. Cho, and we'll be going through chapter 18 on heredity today. Okay, so let's start by talking about the learning outcomes for this lesson. Firstly, we need to understand monohybrid inheritance, including the terms F1 generation and F2 generation. Explain the terms dominant and recessive. Explain why observed ratios often differ from expected ratios, especially when there's a small number of progeny. Alright, let's take a look at this picture over here. So, let's look at this father and his son. What do we notice about the two of them? Right, we notice that there are some differences and there are some similarities. Right, first of all, you can see that the father has straight hair, right? Rather straight hair. Probably he put some gel to make it a bit wavy, and the son has quite similar hair to him also. Right, so hair is something that is transmitted through the genes from our parents. And what about earlobes? Okay, you notice that there are two types of earlobes, attached and detached. Okay, I can't see very clearly from here, so let's just take a quick look on Google. What is the difference between attached and detached earlobe? Now you can go to a mirror and you can take a look for yourself, right? What kind of earlobes do you have? Okay, do you have a detached earlobes like this? That means that you have some extra skin, like some skin dangling below, or are your earlobes completely attached? Okay, so just go and check it out. So there are many differences, right? But there are also many similarities. And ever since, people could observe, everybody sort of understood that there's something that's being passed down from your parents to you. And that is called inheritance. So what is the meaning of inheritance? It refers to the transmission of traits from one generation to another, right? From your parents' generation to you. So there was this man called Gregor Mendel. He was a monk and he worked in a monastery. A monastery is like a church, right? Now, one thing about him was that he was also a very studious person. Right? He was very observant and he would often record down his observations. Now, he was in charge of the garden. And so one of the duty was to grow peas, right? For their soup and for their food. And he started to take records of certain traits that he observed as he was growing the plants. He took note of the tallness and the shortness of the plant, whether the plant had red or white flowers, which plant produced seeds that were yellow, green, round, or wrinkled. And over a period of time, he started to do some experiments. Now, Mendel, he started with this thing called a purebred variety of pea plants. These purebred plants, they will fertilize themselves, right? And then they will produce offspring that will resemble their parents. Okay, so if you recall in our chapter on plant reproduction, right, our gametes, right, from the male parts, the anthers fuse with the female parts in the ovary, and then you form the plants. Okay, and all these plants are what you call as the progeny. So what did he do? Let's look at this experiment. Now, the first thing that he did was that he took some tall plants and some dwarf plants, and he crossed them together. Cross means that he cross-pollinated it. He took the pollen grain from one plant and he put it on the stigma of the other plant. Okay, then he allowed it to germinate everything and then form into a new plant. So when he planted the seed of that cross, he observed that the traits of all of these offspring were tall. And so he called them the F1, which is also known as the first filial generation. The word filial means just like filial piety, right? It means a child, the first children. So what did he notice? He got some plants, he makes tall and dwarf, but all became tall. So that's something interesting, right? So after that, he did it again. He did it one more time. He then crossed this particular tall plant with another one of it, right? And what did he get? This time he got a different variety. He got 3 tall plants to 1 dwarf plant. Okay, of course he didn't just get 3 to 1. He got many thousands, but the ratio was 3:1. So he got a ratio of 3 tall is to 1 dwarf plant. So why did this happen? This was because of the genes that was found inside the DNA of the plant. So he decided to call the trait that was more obvious or more dominant as the dominant trait, right? So that because it's 3 tall, he called it the dominant trait. And he called the other trait as the recessive trait, because this was less seen, right? Less of it, so he called it as recessive. He then started to record down the number of the seeds that was having a certain kind of trait. Okay, and there are many different traits that he observed. Firstly, he checked versus round versus wrinkled. Round versus wrinkled, he noticed that there were about 5,474 round and only 1,850 wrinkled, which is a ratio of near 3:1. He repeated this for various other properties, like yellow versus green seed, purple flower versus white flower, inflated pod versus constricted pod, green pod, yellow pod, axial flower, terminal flower, long stem, short stem. And what did he realize was that across almost all of these different traits, he then got a ratio that is very, very close to 3:1. Not all of them are 3:1, of course. As you may notice, some of them, which have a smaller amount of plants, like for example this one is only 400 and only 150, it was not exactly 3:1, it was like 2.8:1. And we'll talk about that later. Why is that the case? Right? Because these numbers are quite small. That's why I call it a small sample size. Whereas once he had very big numbers, right, in the thousands, then it's going to be very, very close to that ratio of 3:1. So, what did Mendel expect to see? He wanted to see this 3:1 ratio, right? That's what he called as the expected ratio, which is basically what you are hoping to see. But did he always observe this ratio? No, he did not. Why? This was because of the number of plants used. When the number of plants used is very large, we will expect to see a 3:1 ratio. And we might observe this also. We might observe the ratio as near to 3:1. But when there's only a small number of plants being used, the observed ratio of this generation will not be so accurate. This is because of statistics. When the statistics show that when the sample number is very small, the ratios will become inaccurate. Right? This is because of chance and probability. And this is the random nature of fertilization. So basically it's like this, right? You think of a coin. You flip the head and tails. What's the chances of getting a head and a tail? 50-50, right? But if you were to only flip this coin maybe 6 times, will you always get 3 head and 3 tail? No, you will not. Right? Sometimes you'll be going to be 4 heads and 2 tails. Sometimes it'll be 5-1. It's really based on chance. Okay, even though it's you wanted it to be 50-50. But then if you flip that same coin maybe 1,000 times or 2,000 times, that's where you start to see the ratio start to approach 50-50. Okay, so this is the reason why the observed ratio differs from expected ratio. Okay, so this was Mendel's model of heredity. He understood that there was this thing called genes. Okay, so what is a gene? A gene is a sequence of DNA nucleotides that control the formation of a single polypeptide. Now you may recall that in the previous chapter on molecular genetics, we learned about chromosomes, right? We said that chromosomes are basically this tightly wound DNA wrapped around a histone. So if you stretch out the entire length of that DNA, there will be certain sequences, short areas of it, that are called genes. These are the useful areas. They are a sequence of DNA nucleotides that control the production of a single polypeptide. Okay? Let's now talk about how these genes can be separated. During gamete formation, right, gamete refers to the sex cells of the plant. These gametes will contain one pair of the gene. Okay, so this is what will happen. The plant has chromosomes from the father, and the plant has chromosomes from the mother, right? But when they produce the gamete, it will separate, and they only have one of that chromosome, right? So here we have the dwarf chromosome. Sorry, not the dwarf chromosome. This is what the dwarf gene. This is the dwarf gene. This is the tall gene. This is the tall gene. Now you mix them together. You do this thing called a Punnett square. Okay, so this data I'll teach to you. This is called a Punnett square. Basically, you're just like in math, just combining these things together. So you got tall dwarf, then tall dwarf, then tall dwarf, then tall dwarf. Okay, so this will be the first generation, right? We call it as the filial 1 or F1 generation. And what you see here, that all of them are the same. All of them are the same. So that means that they have to be either tall or dwarf. But which do you think is dominant and which do you think is recessive? That's right, the tall is dominant. And so what we see is that all four of these, or four of these, are all going to be tall. Okay. Okay, can take some time to read this. Let's look at how does it work. Okay, we'll come back more about this thing in the next page. Okay, so why does that happen? This is because of the concept of alleles. Okay, so our body has two sets of chromosomes, one from each parent, and each chromosome is represented as a pair in every cell. Now we have learned that genes are this thing called the heredity factors that are found in chromosomes, and they are responsible for the passing down of traits. However, these genes can have different forms. Let's take a look at this example. Here we have a chromosome in blue, maybe let's say it's from the father, and the one in pink is from the mother. They are exactly the same chromosome, right? You can consider it as a homologous pair. That means that they are the same number, right? So like the chromosomes, they all pair up together. So this chromosome, they may be the same chromosome, but there's different variations between them. And let's say, for example, eyelid, right? Eyelid is controlled by our genes. So in humans, we either have the allele for a single eyelid or for a double eyelid. One of them is going to be dominant, and the other one is going to be recessive. What do I mean by dominant? By dominant, I mean that if you have at least one of these alleles, you will then observe this trait in your face. Okay? And if you have neither, then you would get the recessive allele. Okay, so which one is dominant and which one is recessive here? The single eyelid is the recessive one, and the double eyelid is the dominant one. Okay, so if this particular person has these two alleles, double eyelid, single eyelid, this allele will be dominant, and then you will express that trait in that person. So that person will have a double eyelid. Okay, you know what's the difference between double eyelid and single eyelid, right? Let's just take a quick look. Double eyelid, single. It looks like this. Okay, that's this one. Okay, so the difference is in this area over here. This is single eyelid, this is double eyelid. Okay, and you might even be aware of maybe in Korea, right? In Korea, some of the K-pop or their movie stars, they actually go for this thing called the double eyelid surgery because it's to them it's prettier or something like that. Okay, so anyway, double eyelid is dominant, right? Single eyelid is recessive. So actually the picture was right here. Okay, so the recessive allele can only express itself when the person has recessive alleles have two recessive alleles in his genes. Okay, and so a person with two alleles for the single eyelid will have single eyelid. Okay, yeah, they're repeating itself. Okay, so basically, if you see somebody with single eyelid, that means that their parents has passed down the alleles from both parents, father and mother. Both of the genes in that person are single eyelid. Okay, now how do we represent the allele? We represent them using the first letter of the dominant allele. Okay, so you notice here, right? Dominant allele, we use big letter D, and recessive allele, we use small letter d. Okay, we don't use the word R. We don't use the one R. Okay, because we just follow the first letter of the dominant allele. Okay, so like for example, let's look at some examples here. Double eyelid, we use the letter big D and big D. You notice that these are two of the dominant alleles. When you got two of the dominant alleles, you will now call this as a homozygous dominant. The word homo means the same, and zygous just refers to the genetics. Okay, so if you say homozygous, it means same. And you say dominant, it means two dominant genes. If you see this, this person has double eyelid, but in his genotype, it's one dominant and one recessive allele. What does that mean? That means that this person is called heterozygous. So heterozygous means, hetero means different, right? Not the same. So if you have one dominant and one recessive, you are a heterozygous. Lastly, this person is single eyelid. His genotype is small d, small d. Now you notice that they are still using the letter D. They don't use the letter S. Okay, so we're just using the first letter of the dominant traits. Okay, so what is this called? What is small d and small d called? We call this as the homozygous recessive. Once again, homo, it means same. Recessive means the recessive gene. Okay, so let's talk about these two words, genotype and phenotype. The genotype is the combination of the alleles, right? It could be big D, big D, small d, or small d, small d. That's called the genotype. Then what we see, or the outward appearance and trait, is called the phenotype, which is what is observable, right? Our eyelids, our hair color, eye color, these are all observable phenotypes. Okay, so to recap, we have just learned these few new words, homozygous and heterozygous. Homozygous refers to having two identical alleles of a particular gene. For example, big D, or small d, small d. If it's big D, we call that as homozygous dominant. If it's small d, we call it as homozygous recessive. Then, heterozygous means having two different alleles of a particular gene, right? Big D, small d. Okay, so what must we take note of here is that alleles are always controlling the same traits, right? Eyelid is the certain trait, right? We all have only that eyelid. Okay, so what about other things, right? Maybe your blood group is the same kind of trait, but there's different forms of it, right? A, B, and O. There are 4 different forms, so there are different variations, but it must be the same category. Okay, so let's look at some diagrams. Okay, now remember just now we were talking about these plants over here, right? These plants. What was happening actually in the background? What's happening in the genes? What was in the genes of this tall and this dwarf? What's in the genes of this tall? Right? It's actually not the same. This tall is not the same as this tall. There's a difference. So why is that the case? It's because of this thing called genetic diagrams. Okay, what are genetic diagrams? Genetic diagrams are simply things that we use to show how the alleles are being passed down from the parents to the offspring. Okay, so these are what you call as the purebred plants. That means that they're basically homozygous dominant or homozygous recessive. So what happens is that when this plant produces the gametes will always be the dominant allele. Whereas for the dwarf, it will always be the dwarf allele. We then cross these together, right? So if you see the arrows, I combine this and I combine that. Okay, so I get a F1 genotype of all of them are heterozygous. Right, they're heterozygous, they contain one dominant and one recessive allele. So what will I see as the phenotype? I will see them all being tall, and this is called a F1 generation. Okay, the word F1 not always means that they're always heterozygous or tall. It just means that this is the first generation. Okay, of course it depends on the genetics of the parents. Then Mendel took these plants, he took 2 of them and he crossed them again. So F1 times F1. Once again, the gametes split. This time there was a difference, right? There's one that's dominant, one recessive, etc. Now when we cross them, so this gamete plus this gamete combine together, we get top. This one plus this one. Then we get a heterozygous. Same for this, also heterozygous. Then this is, that's right, is a homozygous recessive. But what do we see here? We see that even though there's differences in the genotypes, there are only 2 phenotypes, either tall or dwarf. And there's going to be 3 tall to 1 dwarf. This is what you call as the F2 generation. F2 generation is the children of the F1 generation. Now this is the breeding experiment, right? So they are breeding the plants over here. This same data, alright, these same things is a little bit messy, right? You see a lot of arrows here and there. There's some chances that we might do this wrongly. Okay, and by the way, we need to learn how to draw these genetic diagrams also, right? So you can practice with the worksheet. Okay, the easier way of presenting these results is actually by doing this thing called a Punnett square. Okay, so obviously who invented Punnett square is this person called Punnett, right? So who invented Punnett square is this guy called Mr. Punnett. So it's an easier way of representing the data. Why? Because we start by representing this parent genotype, okay, which is equivalent to this one over here, the F1. So how do I draw a Punnett square? I start by drawing these boxes over here. Okay, so personally, I will just draw these 4 boxes, right? These 4 boxes. This one on top, I don't tend not to draw boxes for them. Okay, and there's a reason why, because it gets very messy. So I will just start by drawing these 4 boxes, and then at the top of the boxes, I'll write the genotypes, right? Big T, small t, then big T, then small t. Then I will do the combination. I combine this and I combine that to get the genotype of TT. Same for this, you get the genotype big T, small t. Same for this, big T, small t. Last one, the 2 small t. Then I can write the phenotypes as a ratio. I write it down, 3 tall to 1 dwarf. Okay. Now sometimes it may not always be 3 tall to 1 dwarf. It depends on the genotype of the parents. Okay, so we'll do some practice of that also in the worksheet. Okay. Let's talk about the test cross. Okay, so test cross is almost the same as this one over here. Okay, so we need to learn to draw both the test cross as well as the Punnett square. Test cross is a little bit harder though. Okay, so test cross is to find out the genotype of the thing. Okay, so almost exactly the same as the Punnett square. Okay, I said there's a lot more info over here. Okay, so how do we do a test cross? We start by talking about writing down the parents' genotypes in a box, in a square. Okay, so in this case, I have the parent genotype, tall and dwarf. These are homozygous dominant, homozygous recessive. Then I split up the gametes, and then I cross the tests together to get the offspring, all 4 tall. Okay, so these are all heterozygous. I can also try that with parents which are heterozygous and one that is homozygous recessive. Okay, so this one will split, this one also split, and in this case we see something different, right? We get 2 tall, 2 dwarf. Okay, this is because I'm mixing a heterozygous with a homozygous recessive. Okay, let's try this. Take out some paper and then you can think about this. Okay, now this requires some logic. Okay, so you see that there's a example over here. Right? Black hair, this is the square means male, circle means female, and if it's not shaded, it means it's red. If it's shaded, it means black. So you see that there's a dad and mom. They have given birth to 2 daughters. One is red hair, one is black hair. This daughter marries Mr. D and gives birth to Dr. G. Then Dr. G marries Mr. F, and then they have 4 children over here. Now using logic, okay, which do you think will be the dominant phenotype? Okay, if you say black, why? Okay, what's your proof? Okay, the reason is because firstly there are more children that are having the black genotype, and also because of this particular generation over here, where you see all the children are having black hair. So what is the genotype of A, homozygous or heterozygous? Okay, now this requires some backtracking. We start by looking at this. Okay, we start by looking at A, right? We see that A and B cross. Then they have 2 daughters, one with red hair, one with black hair. Now what does that tell us? That tells us that it's impossible for A to be homozygous dominant. Because if A is homozygous dominant, then both of the children will definitely have black hair. So the only way that the A can have a daughter with red hair is if the A is actually heterozygous. Okay, so that means that they have one dominant allele and one recessive allele. And just nice, the recessive allele was passed down from A and also from B to C. Okay, then this C has two recessive alleles, and so that she will show red hair. Okay, what is the most likely genotype of F? Is he homozygous dominant or heterozygous? Okay, so likely lah. Now if you see F, you got 4 children that are being black hair, very likely that F actually is homozygous dominant. You have 2 dominant alleles. That means that no matter what, all the children will definitely have at least one of the dominant alleles, and so all the children will be black. Now, can you confirm that F is homozygous dominant? You can't, right? Because why? It might just be probability, just like luck, right? The chances of it happening actually was 50-50, right? 50% chance of the, if he was heterozygous, that's what I'm saying. If he was heterozygous, it was 50-50 of passing either gene. So it could be just be lucky that, you know, you flip a coin and just nice is 4 heads. Okay, answers are here, and take a look. Okay, so let's pause here. You are given a worksheet, so I want you to try all the way until the Punnett square and the test cross. Okay, you will see all these tables and data for you to try in your worksheet, and then when we come back to school, we'll go through it in person. Okay, so with that, thank you and see you in school. Bye. Oh yeah, remember to like and subscribe. Thanks.